DNA fingerprinting is a way of identifying a specific person rather than identifying a more general trait. It can also be known as genetic fingerprinting or DNA profiling. It is hard to distinguish a difference between two people's sets of DNA, as they could have one or more of the same or very similar traits. DNA fingerprinting uses a specific type of DNA sequence, known as a microsatellite, so they can identify them much easier. Microsatellites are short pieces of DNA which repeat many times in a specific person's DNA. In a certain person, microsatellites tend to be highly variable, making them ideal for DNA fingerprinting. By comparing a number of microsatellites in a given area, someone can identify a person really easily. Families can also be determined by DNA fingerprinting, as no child has parents that his parents do not have, and sections of DNA are passed down through generations. DNA fingerprinting is also helpful in forensic, as you do not need an actual fingerprint, because all DNA is in every cell. All you would need would be a piece of skin or a drop of blood. DNA fingerprinting is also useful in identifying bodies that have been destroyed and are beyond recognition. How DNA fingerprinting works is that a sample contains white blood cells, which are broken open using detergent, and then all the usable DNA is separated from the extracellular material. Next, the restriction enzymes are used to cut DNA into smaller sequences. This is worked by cutting the DNA as a specific sequence, which then produces blunt or sticky ends, and these result in many fragments of different lengths. These fragments are called restriction fragment length polymorphs, or RFLPs. These are then put into an agar-ghost gel, and using gel electrophoresis, the fragments are then sorted according to size. When the current of the electric field is turned on, the negative RFLPs will start to move across the gel to the positive end. The smaller fragments move farther across the gel than the large ones. Alkali is responsible for causing the hydrogen bonds to break, and the DNA becomes single-stranded, and when this happens, it causes the nucleotides to become free, and they will later be used on to pair with probes. The gel is then covered by a piece of nylon and thin paper towels, which are used to absorb extra moisture from the gel, and the DNA fragments get gently transferred from the gel to the surface of the nylon. Finally, radioactive probes get washed over the nylon surface. These probes will join to any DNA fragments that share the same composition. Then, by placing a photographic film on top of the nylon surface, the probes leave marks on the film wherever they attach the RFLPs. Dark bands will then show up where the film is developed, marking the length of the RFLPs that were hybridized. Scientists are then able to read the DNA fingerprint and match it to others. They can do this by placing the X-ray on a light background and comparing the RFLP lengths in the DNA. DNA fingerprinting, however, isn't 100% correct because it can only establish a probability. And even though it is a high probability, it may become lower when dealing with direct descendants because they share a large portion of the examined areas of DNA with the parent. Despite all this, however, DNA fingerprinting is becoming more useful in the line of forensics and will be a help to us in the future.